I'm Fred Dick. I'm a professor of auditory cognitive neuroscience here at UCL, and I'm a director of the Brookbeck UCL Center for Neuroimaging. One of the very exciting things about MRI today is that it allows us to do something that for the last 100 years has meant that we've actually had to image either animals or humans in vivo and then wait until they die to actually measure histologically important aspects of their brain. Now, thanks to developments in MRI, we're actually able to look, for instance, at the quantitative estimates of biologically important substances like myelin at the same time in the same scanning session as we're establishing for instance, the topological relationships between areas of cortex and important aspects of functional organization, for instance, the mapping of auditory frequency across the cortical surface. So in the work that we've been doing, we've actually been able to establish for the first time where primary and secondary auditory cortices are without actually having to cut into someone's brain, as was previously the case. Obviously, this is a real improvement in our ability to understand not just the functional organization of the human brain, but also to understand how it is that people vary normally and abnormally within adulthood as well as across development. My colleague Martina Callahan is now going to talk about some of the challenges related to this approach.
My name is Martina Callahan. I'm the head of physics at the Wellcome Center for Human Neuroimaging within UCL. I'm going to tell you a little bit about some of the difficulties that we have in carrying out the work that Fred previously described. So one of the biggest problems we have is that to get images with high enough spatial resolution so that we can be very specific about our conclusions takes quite a long time because MRI is a relatively slow imaging technique. And what this means is that we're very sensitive to the person moving during that acquisition. That can be a problem because it not only causes errors in the quantitative nature of these markers that we acquire that tell us about characteristics like myelin, but also because they can make the data completely unusable. So one thing that we can do to address this is to use a camera that we put into the scanner, and we use this camera to track where the person is by a marker that we attach to them. If we know where they are, then we can update the scanner so that it tracks them, and therefore we get good image quality. And we've shown that this can be very beneficial for the type of imaging that Fred was describing. However, it can't fix every issue that arises because of motion. So for example, when somebody is moving within the scanner, we have different sensitivity depending on where they are. And so this is something that the camera cannot correct for. And what we would like to do as part of this project is to develop models that allow us to take account of the spatially varying and position-dependent sensitivity of the system so that we can then ultimately correct for this and get even better quality data regardless of whether or not somebody is moving during the acquisition. This is important because some of the populations that we would like to image may have difficulty staying still during scanning. This is particularly difficult for children, who we want to study so that we can better understand development, but also for many clinical populations who might have motor difficulties as a consequence of their disorders. UCL is a great place to undertake this type of project because we have expertise across many different domains, neuroscience, clinical and technical expertise. And in addition, we have wonderful facilities, including multiple research-dedicated MRI scanners and this prospective motion correction system.